Hello everyone. A very warm welcome to you all in my class. Myself Preeti Anand, and today I'm going to teach you a topic, mitosis, meiosis, cell division, which is a very important topic for the NEET and other competitive exam. Like from this small chapter, only three or four topics from this chapter. The number of question will be asked in NEET is 2. The total marks will be 4 plus 4 is equals to 8, and the weightage is 4%. So from this a very small chapter of 3 or 5 pages of a chapter, the number of chances of coming questions will be 2, the 8 marks. So this chapter is very important for you all for the NEET and other competitive exam. And today I'm gonna teach you mitosis, meiosis, cell division. So let us start this topic with a very positive vibe. Also, you are aware that all the living organism, even the largest and the smallest, all starts with the life form from cells. So let us start this interesting topic with me. First of all, I would like to ask you all that what is cell? We all have studied what is cell in class 6 or in class 8 also, in class 9 as well as in class 10. Now it's a topic of class 12. So what is cell? It's a very basic question for you all. So now quickly you can tell me what is cell? Like as we all know that the cell is a basic structural and functional unit of life, and also growth and reproduction are the characteristics of cells. All cells reproduce by dividing into two, and the newly formed daughter cells themselves grow and divide further. Like there is a cell, it will divide and it will form two cells. Now these two cells will again divide and form four cells, and then the replication will go further and the division will take place simultaneously. So after understanding what is cell, we have to understand what is a cell cycle, because it is a basic cell cycle is a very important topic for any living organism. Without cell cycle, there is a no life. So quickly we will understand what is cell cycle. Basically, cell cycle is a very important process for the living organism in which there is a DNA replication, reproduction of cells, regular sequence of growth, and the division of cell. If there is no replication of DNA, then there is a no life. If DNA is replicate and without reproduction of the cell, there is a no life. So these all are the set of processes which requires to live for a living organism. Without all this process, there is a no life form in a space in anywhere. So quickly we will understand different phases of cell cycle. These are the different phases of cell cycle. Let us understand it quickly. First of all, from this diagram, I'm going to teach you the G1 phase. There are four phases like G1 phase, GO phase, S phase, and G2 phase, finally M phase. First of all, we'll talk about the G1 phase, the very primary phase, the very first phase, G1 phase, in which there is a growth phase in which the cell will grow, like it's a primary growth phase of the cell, the period of growth and the preparation for the DNA synthesis. In the G1 phase, there is a no DNA synthesis. The cell will only grow, like suppose this is a cell will grow in the size. It will not replicate, there will be no DNA synthesis, only the growth will be there in the G1 phase. This is a very first phase. In G1 phase, the cell will grow. The cell is metabolically active. I'm not telling that cell is inactive. Cell is active, cell is growing in their size, cell is developing their intercellular components, but it will not divide. It will rest in this phase. It will wait for the next phase, S phase. If the cell in G1 phase, in a resting phase, it will go in an other phase, S phase. In S phase, they will do synthesis and the replication of DNA. So first, I'm sure you all are clear that in G1 phase there is no synthesis of DNA. Only the cell will grow, metabolically active. It will grow in the size, and then it will come on the GO phase. If some cell like a nerve cell is don't want to grow, they don't want to divide, and there will be no synthesis of DNA. So the cells which don't want to replicate, don't want to synthesize their DNA. They just want to be in a resting phase. So there is a geo phase, which is also known as quiescent phase, in which the cell will take a rest. It will not divide like nerve cells, for example. And then S phase, where the main synthesis is take place, preparation of growth and the preparation for the DNA synthesis. In S phase, the synthesis of DNA will be there and the replication of DNA will be there. And then in G2 phase, where the second growth phase, final phase for the preparation of cell division. It is a final phase for the preparation of cell division. After the G2 phase, there will be M phase. M phase is mitosis phase. M phase is known as mitosis phase, and the cell will prepare for the division. And in M phase, the division of nucleus resulting to equal separation of the chromosome. So hope I'm clear that there is a G1 phase, G0 phase, S phase, G2 phase, finally M phase. M phase where the division, where like this is a cell. In G phase, this is a cell. Cell is growing. And the S phase where the preparation is taking place. So the cell is prepared. In S phase, the cell is preparing. DNA synthesis is there for the division, and in G2 phase the cell is completely prepared for the division. And in M phase, what happened? The cell will divide like this. Cell will divide and cell will separate in different phases. Cell will divide and separate. In mitosis, there is a different phase stages like prophase, metaphase, anaphase, telophase, and cytokinesis. We will see all these stages. This is a different phases for the cell cycle. There are also checkpoints. Whether if you are preparing for anything and you will check in more like mock test, you will give a test whether I'm prepared or not, and what is how is my preparation, how is my preparation. So how I check, how we all check how is my preparation for the NEET, for the any competitive exam. We just give a mock test, we just give a test to know how is my preparation. So sometimes we get 50%, sometimes we get 70%, sometimes we get 90%. So this is the way we are checking our preparation. That's how the cells are checking their preparation by different checkpoints. Like first in G2 checkpoints, whether the cell is prepared or not for the division. In the GO phase, and in G1 phase and the S phase, if there is every checkpoints where the cell is checking themselves that they are prepared or not. This is a diagram. This is a very beautiful diagrammatic representation for the cell cycle, indication formation for the two cells from the one cells. Hope I'm clear till. If any question you can ask me. Cell cycle divides in two basic phases. Like cell cycle has two basic phases. Cell cycle has two basic phases. One is interphase and the other is M phase. Interphase, M phase. Cell cycle has two phases, interphase, M phase. In interphase, there are again G1 phase, S phase and G2 phase, which I have explained, which I have given the overview. Only this is the only overview for the cell cycle. In interphase and M phase. So in interphase, again there are three phases. First of all, I'm going to tell you the approximately time taken by the eukaryotic cell to divide. Like eukaryotic cells divide in approximately 24 hours. Sometimes yeast took only 90 minutes to divide, and different cells have their different time to divide. And in interphase, what happened? There is three phases: G1 phase, S phase and G2 phase. Interphase represent the phase. We can also say that the interphase represent the phase between two successive M phase. I will tell you interphase and M phase. Interphase, see, understand it. Interphase and M phase. Interphase represent the phase between two successive M phase. One M phase and another M phase. Like this is first M phase, this is second M phase. The duration between two M phase is known as interphase. M phase, second M phase. The duration between the two M phase is known as interphase. This is also resting phase in which the cell is preparing for the cell division. Interphase is when the cell grow and organelles double prior to the actual splitting of the nucleus, and 93% of the cell life is spent in the interphase. Now M phase. Interphase, M phase, interphase, M phase, interphase, then M phase. In M phase, mitosis phase, the phase when actually cell division occur. The cell will prepare in interphase and division will be in M phase. M phase starts when the nuclear division corresponding to the separation of daughter chromosomes. The separation of daughter chromosome is known as karyokinetics. Direct question come in NEET. Direct question, this is a direct MCQ question, karyokinetics and the cytokinesis. So karyokinetics and usually end with a division of cytoplasm, cytokinesis. What is cytokinesis? Direct question for MCQ. Please write it down. The direct question, karyokinetics, cytokinesis, these all comes in the previous year questions of a many competitive exam. This question have come, cytokinesis. Now interphase, M phase. Interphase has again three phases. Growth phase, synthesis phase, and growth 2 phase. First, growth phase. S is DNA synthesis, G2 is growth and preparation for the mitosis, M is mitosis cell division. Now interphase, the very first stage is G1 phase, growth 1 phase is, I have already explained you, the growth 1 phase is when the organelles double. Cells are metabolically active, continuously grow in size but does not replicate their DNA. Cells grow in size but does not replicate their DNA. S, when DNA is replicated and the synthesis of DNA. During the time DNA per cell doubles, each cell needs a complete and identical set of DNA. Now in G2 phase, the final stage for the growth of the cell. Protein needs for the mitosis are produced and the cell continues growth but does not divide. The cell will divide only in S phase. The cell will only divide in the M phase. S is the synthesis time. So these are the previous year question. I took only 2 questions here. This is a previous year question. DNA synthesis take place during which phase? You have to tell me. DNA synthesis take place during S phase. Synthesis S. S for synthesis, S for S phase. In S phase, synthesis. S phase is for synthesis. S phase, synthesis phase. Centrosome duplication take place in the phase S phase. Mitosis. Now after interphase, these all about interphase. G1, S, G2, G0. This is all about the interphase. Now come to the M phase, where prophase, metaphase, anaphase, telophase, cytokinesis will be there. The mitosis occurs in somatic or body cells. Example, skin cell, nerve cells. The process by which the cell nucleus divide into 2 identical nuclei. In some humans, interphase lasts for 15.3 hours, while the mitosis took only for 0.7 hours. Occurs in a series of steps. These are the different steps for the mitosis: prophase, metaphase, anaphase, telophase, cytokinesis. Quickly understand this: prophase, metaphase, anaphase, telophase, cytokinesis. These are the overview for all these processes. I hope your interphase and mitosis phase will be clear. Any question you can ask me. Interphase is clear, right? G1, S, G2, interphase. M phase, mitosis phase. Prophase, anaphase, telophase, cytokinesis. Prophase, metaphase, anaphase, telophase, cytokinesis. Fine. So this is a diagrammatically representation for the interphase. This is prophase, metaphase, anaphase, and telophase. One of a previous year question. Let it do it quickly. If you want to count the number of chromosomes, which is the best stage to do so? This is your homework. If you want to count the number of chromosomes, which is the best stage to do so? This is M phase. So this is a chromosome. What is chromosome? You have to tell me what is chromosome. Must duplicate and separate during mitosis. Structure a tightly packed DNA. DNA is tangled up into a substance known as chromatin. The chromatin is packed on the chromosome. This is a cell, and this is a nucleus inside the cell, and the chromosome telomere. This is a tangled form of DNA. So okay, prophase. In M phase, the very first stage is prophase, where what happened? See, the chromosomes now called chromatids. The chromosome which are present inside the nucleus. The chromosome now called chromatids because they are doubled to form the short, thick rods which pair up and line up in the center of nucleus. A centromere connects to two halves of the double chromatids, and the spindle fibers. Look, this is a cell where what happened? The spindle fibers begins to form. These are the spindle fibers. Spindle fibers play a very important role for the separation of these chromosomes. Spindle fiber begin to form. And what is spindle fiber? First of all, what is spindle fiber? A fibrous structure from the cytoplasm which forms to the centriole. What is centrioles? Centrioles moves to opposite side of the cell. Centrioles move opposite side of the cell. These play very important roles in the separation of the chromatids, sister chromatids, and the nuclear membrane breaks down. If the nuclear membrane will not break, then how replication will take place? So the breakdown of nuclear membrane is very important for the further replication. This is a cell. This is the cell. Inside this there is a nucleus. If inside this there is a nucleus, inside cell there is a nucleus. If the nuclear envelope will not break down, how the genetic material will release out and how there will be replication? So the breaking of nuclear membrane is very important process which will take place in the prophase. This is a diagrammatic representation. Like see, there is a chromosome, spindle fiber preparation. Spindle fiber is forming nucleolus, astros, centromeres. These spindle fiber connected with the centromeres, kinetochore, and nuclear envelope fragments. See, this is the fragments of nuclear envelope. The breaking of nuclear envelope is started. Centrosomes connected with centrosomes. Previous year questions. Individual chromosomes become distinct through the light microscope during the mitotic stage is prophase. Nucleolus and endoplasmic reticulum start disappearing at, this is the previous year NEET questions. This question comes in AIMS. This is the previous year, very important, very important questions. These are very important diagrams. Please try to pay attention in this diagram. Only 3 topics and 2 questions. You can imagine how important topic is this. Nucleolus and endoplasmic reticulum start disappearing at early prophase. Now after completing prophase, what happened in M phase? M phase is metaphase. Prophase, metaphase. Metaphase is complete disintegration of nuclear envelope. What happened in prophase? Prophase means in prophase there was a breaking of nuclear envelope. Now in prophase there is a complete integration of nuclear envelope. The nuclear envelope is completely integrated. Now the material from nucleus will release out. Centromeres of the chromatid pairs line up in the middle of the cell. This line. The centromeres from the chromatid pair line up in the middle of the cell. They will lie up like this. They will lie up in an equatorial plane like this, and then they will separate. They will lie up, first of all lie up, and then they will separate. They will lie up on middle of the cell. This is known as metaphase plate location, where the centromeres lie up in the center of the cell. By the end of metaphase, the chromatid has attached to the spindle fiber. See, this is a pole of spindle. You can see this is a pole of spindle pole. Metaphase plate is there, the polar fiber, nuclear envelope fragments. See the fragments. The nuclear envelope is completely breakdown. See the fragments. Kinetochore fiber is there which attach, kinetochore fiber which helps to attach, and the pole of a spindle. This is a metaphase. So these are the previous year question. Quickly we have to do it very quickly. The stage in which chromosomes align on the equatorial of the spindle fiber, tell me, equatorial, plane, the metaphase plate. In which stage the metaphase plate is formed, is metaphase. The best stage at which the total number of chromosomes